enzymes, no? Biomedical importance. Of course, you really need your enzymes to break down the food that you eat to become nutrients, and those nutrients supply as energy or building blocks for parts of your body, you know, like into proteins, into DNA, membrane, cells, tissues, and responsible also for your locomotion, nerve function, and muscle contraction. Now, all enzymes, almost all enzymes, are proteins. It is just a few years ago that the researchers came up with enzymes that are not proteins, and we call them ribozymes. They are usually related to the RNA. In fact, mitochondria is one big ribozyme. No, I'm sorry. It's not the mitochondria. It is your ribosome. Sorry for that. Okay. Enzymes are effective and highly specific catalysts. So enzymes act on the substrate and they form, they modify or they produce what we call products. Okay. Now, it says here that they are highly specific. Yes, enzymes are highly specific, but really not all are highly specific, because there are enzymes that can have several substrates.
Yes.
Okay. All right, just to show you this 3-point attachment. On this side here we have the enzyme. Okay, and here is the substrate. Now, specificity provides that when it is attached to the substrate, there's no other way that it can attach. Like, for example, the numbers here, the numbers here, 1, 2, and 3 on the enzyme. This is the part of the enzyme that is attaches the substrate. So the substrate, as you can see it here, this one attaches to that one, and this 3 attaches to number 3, and number 2 attaches to number 2. So that's a 3-point attachment. Now, if you turn this around, there can be it cannot fit. No? So it cannot fit. So it is really specific because it requires a 3-point attachment. Now, if you look at this, 1, 3, and 2 can fit snugly on the enzyme. And now, if you turn this substrate around, there is no way that you can have what we call a 3-point attachment. Okay? All right. Enzymes are classified by reaction type. Enzymes are grouped into following 6 classes: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. On this side here, you have the mnemonic for that, OSLEL. So oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases. Okay. Oxidoreductases, from the name itself, you know that it is to catalyze oxidation and reduction. Transferases, from the word transfer, so these are the enzymes that catalyze transfer of groups or moieties, no, to another recipient of transfer. And we have hydrolases. These enzymes catalyze cleavage or the break of bonds, carbon to carbon, carbon to oxygen, carbon to nitrogen, and other covalent bonds. Okay, hydrolysis. And it uses water to break this. Lyases also can do the same thing as a hydrolase, but the only difference is that lyases does not use water to break the bonds. No? And the reaction of this lyases after the catalysis, after the enzymatic reaction, they usually leave behind a double bond. That's the lyases. Then you have your isomerases. So this would catalyze, or reactions here would be more of a transformation from one isomer to another. No? So it's geometric or structural changes within a molecule. Ligases comes from the word ligate, meaning to tie. Enzymes that catalyze the joining together of two molecules, coupled or utilizing energy, and that the use of energy, ATP, is involved. Okay, so it is easy to remember. The 6 classes of enzymes. Now, there are so many enzymes in the body, thousands of it. So it's very hard to remember all of them. But what we can do is just remember what their actions are. So that's why we have classified the enzymes into reaction type, and there are only 6 of them. Oxidoreductases, we have enzymes for oxidation reductions. Transferases, enzymes for transfers of groups or atoms. And hydrolases, enzymes that break bonds utilizing water. Lyases, also the same, can break bonds, covalent bonds, but it does not use water. Isomerases, interconversion of isomer, okay, and then ligases for tying down 2 molecules together and utilizing energy in the form of ATP. All right. Enzymes, we have the prosthetic groups, cofactors, coenzymes. They play important roles in catalysis. Many enzymes contain small molecules or metal ions that participate directly in the substrate binding or in the catalysis. They are termed prosthetic groups, cofactors, or coenzymes. Okay, so we have prosthetic groups, And we have cofactors and we have coenzymes. Prosthetic groups are non-protein in nature. Non-protein. Cofactors with associate reversibly with enzymes or substrates. And then we have coenzymes. Okay, we will be able to see that. Many coenzymes, cofactors, and prosthetic groups are derivatives of vitamin B or B vitamins. You know, vitamins, if you recall, there are two classes. You have the water-soluble vitamins and
Not soluble.
the non-soluble in water vitamins. It's easy to remember.
Addict.
For the non-soluble, you have
Addict.
the mnemonics there that you can use. ADEK, A-D-E-K. Oh, A-D-E-K. Vitamin A, vitamin D, vitamin E, vitamin K. And then you have your soluble vitamins. There are only two kinds. Vitamin C and the vitamin B vitamins. Now, vitamin B, there are so many. B1, B2, B6, B12, B3, B5. Okay, so we will be using them. All right, now, water-soluble vitamins. Okay. Now, a vitamin nicotinamide, derivative of nicotinic acid. Nicotinic acid, sometimes you might think it comes from nicotine. It does not. Nicotinamide. Okay, and you have two of those NAD and phosphorylated NAD, or NAD and NADP. So, nicotinamide, adenine dinucleotide. And this one is nicotinamide, adenine dinucleotide
Phosphate.
phosphates. Now, this nicotinamide reacts with a derivative of adenine. Adenine dinucleotide or adenine. No? So we have nucleotides here involved. Adenine dinucleotide. This one is with adenine with a phosphate. Adenine dinucleotide phosphate. So that's, this can come from niacin. Remember the B vitamin family. Then we have riboflavin, vitamin B2, the coenzymes, protein mononucleotide. So again, nucleotides are involved. And then we have flavin adenine dinucleotide. And then pantothenic acid, which is part of the formation of coenzyme A. Coenzyme A. Or CoAs. And then we have thiamine, vitamin B1. which the coenzymes is thiamine pyrophosphate, used in decarboxylation of keto acids, while folic acid and cobamide, vitamin B12, coenzymes function as in metabolism of one carbon. There are several other coenzymes. We will be dealing with them later. Now this one is your this is your NAD. Okay. This is your NAD. Whole thing there. Now this OR here, this is where you put your phosphate and it becomes NADP. No. All right.
Right.
So coenzyme serves as substrate shuttle. Now usually reactions, it does not really mean that when once a substrate is attached to the catalytic site of the enzyme, that there will be reactions right away. In fact, there are what you call relay of electrons or protons that have to be transferred. So as we said here, analysis sprung from observation. The presence of substrate rendered enzymes more resistant to denaturing effects of an elevated temperature. This observation led Fisher to propose that enzymes and their substrates interact to form an enzyme substrate complex, or an ES. So before a substrate can be transformed into a product, the substrate must first bind to the enzyme. As what we have shown you there in a previous slide, the 3-point binding. In the ES complex, catalysis, of course, and then products are formed. Fisher is he says that high specificity with enzyme, which enzyme discriminates their substrates when forming an AS complex, was analogous to the manner in which mechanical lock and key, meaning you need to have the right key to open a lock. The analogy to enzyme is that the lock is formed by a cleft or a pocket on the surface of the enzyme called the active site. Now, I do not want to use this active site when you refer to the catalytic site, because actually in an enzyme, especially allosteric enzymes, there are more than one active site. Okay. So when you refer to the site where catalysis is occurs, do not call it active site. Call it catalytic site. As implied by the adjective active, an active site is much more than simply a recognition site for binding. It provides the environment wherein chemical transformation can take place. Within the active site, substrates are brought into close contact, no, or optimal or correct alignment with the cofactors and prosthetic groups and the amino acids that participate in the catalysis of the substrate. And furthermore, it says here that the catalysis is further enhanced by capacity of the active site to shield substrate from water, because water can affect the catalysis and generate an environment whose polarity, hydrophobicity, acidity, or alkalinity can differ markedly from that of the surrounding cytoplasm. Okay? Two-dimensional representation of a dipeptide substrate, glycyl tyrosine bound within the active site of carboxypeptidase. Okay, so you have here the tyrosine. Here. And it is bound now to this cleft over here. This one. See that? Okay. Enzymes employ multiple mechanisms to facilitate catalysis. Catalysis by proximity. So, catalysis by proximity, it means that the substrate and the enzyme should be very near each other. Otherwise, nothing happens. So it has to be very, very close to each other. And then you have your acid-base catalysis, where photons are involved, the breaking of the bonds. And then catalysis by strain, wherein one part of the substrate is being pulled by a strong force brought about by bonding with the catalytic site, and on the other side it is being stretched out by a bonding on that particular side on another portion of the catalytic site. Then covalent catalysis. Okay. This is the breaking bonds of covalent. This is breaking of covalent bonds. Now what is shown here is ping-pong mechanism for transamination. So we have here an enzyme and a molecule, and alanine. So these two have to react with each other. Okay. All right. So give us the two this compound, an alanine, must be bound to the enzyme. The enzyme is here. So you have this compound and you have here alanine. Right? And then reactions occur in the catalytic site. Okay? The amino group of alanine is transferred to this compound. So what is left of the alanine is the carbon skeleton, and that is pyruvic acid or pyruvate. So there you are now. You have an amino group attached to a methyl. All right. Next reaction would be to remove this pyruvate. Pyruvate is removed. This compound is still attached to the enzyme over here. And then alpha-ketoglutarate. Alpha-ketoglutarate is the carbon skeleton of glutamic acid. What do you mean by carbon skeleton is the organic acid of the amino acid. So which means that this is a carbon skeleton, alpha-ketoglutarate. There is no amino group here. If you add an amino group to alpha-ketoglutarate, it becomes glutamic acid. Becomes glutamic acid. So you bind the alpha-ketoglutarate to the enzyme. So they are both now here in the catalytic site of the enzyme. And then this amino group is transferred to alpha-ketoglutarate and becomes glutamate. Okay? And you have that compound, and then glutamic acid is released, and then you still have that compound there starting from here. Now, what have we transferred? We just transferred amino group coming from alanine. It is transferred to this compound. So you have there the temporary lang. Then the pyruvate is removed. Okay. And then alpha-ketoglutarate is catalytic site. This amino group now is transferred to that and becomes now glutamic acid, and you have that compound there. Glutamate is released. So, in short, the amino group of alanine has been transferred to alpha-ketoglutarate to become glutamic acid. Correct? So that is just transfer of an amino group. So we call that a ping-pong mechanism. No? Just like the ball is transferred from one part of the table to another table and all. Okay. Now this one shows binding of the binding of substrate to the catalytic site. There are two kinds. One is Fischer, and it is called lock and key theory. Theory by which that the shape of the substrate would be similar to the shape of the catalytic site, so that it can fit in. Okay, so that is Fischer. or what we call the lock and key theory. Now here we have Husland's theory. Husland's theory is known as the induced fit theory, where compound is attached and attached here. Look at this. A to this portion of the catalytic side, B into this portion of the catalytic side. Okay. Now, as a result of the binding, some bonds are broken, so that whole enzyme now changes its shape. See, it bends. Yes, it bends like that. As it bends, creates a strain here, and the bond is broken. So the binding of A and B, AB, creates a conformational change. Change in shape. Changes its shape. And that is Husland's theory. The other theory is Fisher's theory, lock and key. This one is known as acid-base catalysis. So protons and electrons, no, all right, you have aspartic acid, call it Y, and then aspartic acid, where this compound attaches itself, and you call it now X. And then there is transformation, and the aspartic acid is released to the original. So you have aspartic acid Y. And also the aspartic acid that has been transformed with an OH, the carboxyl group is released here, that's bound, and then you have restore this aspartic acid X. Okay, and then this loses its protons, and then it becomes the original aspartic acid Y. Okay. Now, here, where you have a transfer of electrons, then we have it here, so that the proton now is released, so that you'll have an OH. Okay, and you have this compound now having an OH, which you do not have it here. So we call this enzyme a protease acting on a protein, which we call an aspartic protease, because the active amino acid in the catalytic diet is aspartic acid. So it's called aspartate protease. There are other proteases. A very common one is serine protease. So the active amino acid is serine with its OH. Okay. This is a charge relay system. Removes the proton and then serine 195, the OH of serine, the proton is removed. No? This one here, catalyzed by histidine 57. So it's charge relay. So many. Okay. So we say activated serine attacks the peptide bond, forming a transient tetrahedral intermediate and release the amino terminal peptide. It's facilitated by donation of a proton to the newly formed amino group by histidine 57 of the charge release system, yielding an acyl serine 195 intermediate. Histidine 57 and aspartate 102 collaborated to activate a water molecule. Okay, water molecule which attacks acyl serine here residue 195. Okay, so that's how it is. No. And the charge release system donates a proton, facilitating down of the tetrahedral intermediate, and to come up again restoration of your serine 195, histidine 57, and aspartate 102. Okay, this enzyme, fructose 2,6-biphosphatase, a regulatory enzyme of gluconeogenesis, a pathway by which glucose is formed from non-carbohydrate source, which catalyzes hydrolytic release of the phosphate on carbon 2 of fructose 2,6-biphosphate. Figure 7-8 illustrates the roles of seven active site residues. Involves a catalytic triad consisting of 1 glutamic acid and 2 histidine residues. There you are. This is the catalytic site as the substrate. Okay. Fructose biphosphatase. have carbon 2 of fructose, carbon 2 of fructose, and carbon 6 of fructose contains phosphate. And the phosphate are being removed. There, see that, being removed. Hold for a while by histidine 258 and then release as an organic phosphate. So that is how a compound fructose with 2 phosphates and a phosphate. Catalytic residues are highly conserved. What do you mean by catalytic residues? These are the amino acids found on the surface of the catalytic site. These are known as catalytic residues. And in the formation of the protein, the catalytic residues there, which are utilized for catalysis, are highly conserved in the genetic code, meaning they do not change so easily. No, it may take several thousand years or before it can be changed, or unless there is something, a genetic manipulation by human beings, uh, which is now, of course, possible. So if you just leave it to nature, the catalytic residues there, the amino acids involved in catalysis are highly conserved from one species, no? And from the particular species down to its siblings. All right, what are isozymes? Now, these isozymes are very, very important enzymes, especially for diagnostic purposes of a disease. Isozymes are distinct enzymes, so they are not the same physically and chemically, but they have the same catalytic reaction. They have the same catalytic reaction. All right, most of these are your LDH, no? Lactate dehydrogenase. We have LDH1 up to 5. The good thing about these isozymes are they are located in not in one particular organ. They are dispersed. They are dispersed. So, if you can identify the particular isozyme that is in the blood, then you'll be able to know which organ is in a diseased state. Okay. Question now is, why do these isozymes go into the blood? Okay. These isozymes go into the blood when the cells dies, because everything the cell content, as it dies, it goes into the blood. Then you'll say, what's the connection? Well, when the cell dies, it breaks. So where is the content? of the content of the cell that died. It goes into the surrounding tissue. And what do you call that fluid that surrounds cells?
Interstitial fluid.
All right,
ICF.
interstitial tissue or interstitial fluid. Okay. Now, where will this interstitial fluid be removed? Because it contains materials from a dead cell. How is it removed? No idea?
Isozymes.
Are you familiar with lymphatics? What are lymphatics? The lymphatics drain the interstitial fluid or the interstitial surrounding of the cells. So when it drains that, the lymphatics drains that. Lymphatics also has tubes like blood vessels. So it drains that. And this lymph, the lymph, you call it the lymph inside the lymphatic vessels, connects to the general circulation, or to the blood circulation. Now, lymphatics from the feet to the thighs, or lymphatics around the abdominal cavity, they coalesce and form a big lymphatic duct. And that lymphatic duct goes up, penetrates the diaphragm, and goes up to the left subclavian vein. Left subclavian vein is in your shoulder, left shoulder, around there. And then it connects to the left subclavian vein. So that means the lymphatics goes into the blood. Diba? So when it mixes in the blood, all the contents of the dead cells, where it came from, are there. And it can be detected by enzyme assay. And we said that these isozymes, like for example, LDH has 5 isozymes. And these isozymes are not located in one organ. So LDH1 will be another organ, and LDH2 another organ or another part of the body, and so on. So if LDH1 location in a particular organ and the cells there in that particular organ are destroyed, so all of its contents goes into the blood or into the lymphatics, into the interstitial tissue, then it goes into the lymphatics, and the lymphatics connects to the left subclavian vein. And then it mixes with the blood. So if you do an assay on the blood and you find LDH1, so you know which organ is diseased, or there are cells that are destroyed there. Okay, so that is how you do an enzyme assay. You must have remembered, especially the medtechs, the nurses, that there are 2 kinds of enzymes in the blood. 2 groups of enzymes. Enzymes that are present in the blood for the functioning of the blood. A very good example of those enzymes are the enzymes for formation of blood clots. So those enzymes are always there. So those enzymes are not good for detecting diseases through enzyme assay. And so the other one are the enzymes that are not supposed to be there in the blood, but they are forced into the blood because the cell dies and it empties itself into the interstitial tissue or fluid, and it is drained by the lymphatics, which brings it to the general circulation. So enzymes that are not supposed to be there are enzymes that have to be detected through enzyme assay, and you'll be able to know what particular organ has a problem. Okay? You know now the reason for that enzyme assay. So catalytic activity of enzymes facilitates their detection. So when you do a normal, let's say, you want to find out if this particular enzyme are present. So what you do is because enzymes, the presence of enzymes are not detected right away unless there is the product. So one way of detecting enzymes or activity of the enzymes, because you cannot, how can you determine the activity of an enzyme? No, it's very hard. It's the activity that you are going to assay. So what you are going to do, you are going to assay the products that are formed of the enzyme. So if you and the products formed are more or less equal to the amount of the enzymes. Because one enzyme, one substrate equals one product. Right? All right. Never mind this. Oh, this one. Dehydrogenases. So enzymes known as dehydrogenases, from the word dehydro, so dehydrogen, meaning you remove hydrogen. Okay? So you're actually removing hydrogen from that particular substrate. And usually, when you have dehydrogenases, the hydrogen that has been removed is transferred to a temporary what you call acceptor. And dehydrogenases uses acceptor, the coenzyme NAD or NADP. NAD or NADP. So you can assay the activity of the enzyme by measuring the products that are formed. And in this way, when a dehydrogenase remove a hydrogen, the temporary acceptor of hydrogen is NAD. So NAD plus a hydrogen, it becomes NADH. All right. Now, What is shown here is this one. You will notice that NAD without the hydrogen, no, the light absorption is very low. Diba? But with NADH, it is very high. There, around here, 0.3, somewhere. While your NAD only without the H is almost zero. At 350 nanometers. So optical density. Okay, so you're going to measure this one because this is measurable. This is the one that is measurable, but not the activity of the enzyme. So when you measure this, you are actually, because that is, if there is so much of NADs for, therefore the activity is high. If there is no very low NADH, then activity is low. You get it? So NAD or NADP dependent dehydrogenases are acid spectrophotometrically. So you need a spectrophotometer. Oh. And NADH, once it is reduced, this is oxidized, this is reduced, it's at 350 nanometers. You can detect it in your spectrophotometer. So it says here, many enzymes may be acid by coupling to a dehydrogenase. All right. So we know what is this, no? This one, the presence of NADP, and you'll be able to produce this. Now, if you're interested in seeing the activity of hexokinase, hexokinase is an enzyme that puts a phosphate to a substrate. The substrate is glucose, and a phosphate is added, so it becomes glucose 6-phosphate. The phosphate is added at carbon 6 of your glucose. So where does this phosphate come from? It comes from adenosine
ATP.
triphosphate. Okay, so one phosphate is added, it's removed, and added to glucose at this number 6 carbon, phosphate, glucose 6-phosphate, and your ATP becomes ADP. Now notice this magnesium. The role of this magnesium is to stabilize your ATP or ADP. Why? Because the phosphate here is very, very strong acid and becomes negatively charged. So in order that this will not change the pH, so magnesium with positive charges 2, and you have a phosphate here. So 2 phosphates with 2 negative, then that is being remedied by 2 positive from magnesium. Okay, so as we said, we are going to see the activity of this, but we couple it to a dehydrogenase. And here is our dehydrogenase. And a dehydrogenase here removes the hydrogen from glucose 6-phosphate. Okay, and the acceptor of that is NADP. and becomes NADPH plus H. Okay, and the product is phosphogluconolactone. And in that way, the amount of NADPH that will be formed will be more or less similar to the amount of hexokinase, activity of the hexokinase. The more NADPH are formed, then that tells you also how much is your hexokinase. Okay? So it is being coupled to a dehydrogenase because this one is measurable. This one you cannot measure this. This one is measurable. Okay, this one. So these are the diagnostic assay done in the laboratory of hospitals. So we do assay of ALT or alanine aminotransferase that will tell us about viral hepatitis. If this is high, you will have viral hepatitis. Amount of enzyme amylase and acute pancreatitis. Ceruloplasmin, we have Wilson's disease or hepatolenticular degeneration. Creatine kinase, you have muscle disorders. CK, no? Creatine kinase. All right. Lactate dehydrogenase isozyme 5, liver diseases. Lipase, acute pancreatitis, similar to that, amylase. Then you have your beta glucocerebroside or cerebrosidase, Gaucher's disease. Alkaline phosphatase is another isozyme, bone disorder, and obstructive liver disease. These are just only a few of enzymes used for clinching the diagnosis of the disease. So there is this also analysis of serum enzymes following tissue injury. Very good example of that is when you have myocardial infarction. You have myocardial infarction, so that means a blood vessel is being blocked, and cardiac cells dies, and it empties into the interstitial fluid. The interstitial fluid is being what you call drained by the lymphatics. The lymphatics join with the general circulation, so you can be able to detect enzymes that are present in the cardiac cells, cardiac muscles. Okay. You can detect the presence of LDH1 in here, myocardial infarction. Or you have your CK creatine phosphate, creatine kinase, CK3. Okay, now, so you have creatine kinase, 3 tissues. Okay, CKMM, skeletal muscle. CKBB, brain. CKMB, heart. No? You have LDH and your CK isozymes are separable by electrophoresis. So you have three types of CK. CKMM or skeletal muscle, CKBB is brain, CKMB is heart. So the first enzyme used to diagnose myocardial infarction was AST, aspartate aminotransferase. Alanine, ALT, aminotransferase, and lactate dehydrogenase. Diagnosis using LDH exploits tissue-specific variations in the quaternary structure. However, it is released relatively slowly following an injury. Creatine kinase along with other optimal diagnostic window as with LDH. So LDH is not released right away. This one is a little bit faster than your LDH. This is your lactate dehydrogenase. Normal pathologic patterns of lactate dehydrogenase isozyme in human serum. So in the heart, you can see this pattern here. Normal and in the liver pattern. Normal and pathologic patterns of lactate dehydrogenase isozymes in human serums. So you just separate that by electrophoresis. Your isozymes were then visualized by dicoupled reaction specific for LDH. Pattern A, pattern B, pattern C. The one that we have shown you, this one. This one here. For electrophoresis. Serum, a myocardial infarct, a normal serum, and serum from a patient with a liver disorder. All right, additional uses of enzymes are employed in clinical lab to determine the presence and concentration of critical metabolites. For example, glucose oxidase test utilized to measure the plasma glucose concentration. Enzyme that acts on glucose, oxidizing the glucose oxidase test. Enzymes are also used with increasing frequency for treatment of injury. Example, plasminogen activator, TPA, or streptokinase for treatment of acute myocardial infarction, and trypsin for the treatment of cystic fibrosis. So they are using enzymes. Or you have their streptokinase to dissolve the clot, myocardial infarct. Intravenous infusion of recombinant produced glycosylases can be used to treat lysosomal storage syndrome. So these are all, these are not common disorders. These are genetic disorders. But this one is very, very common. Okay, so enzymes can facilitate diagnosis of genetic and infectious diseases. So they go into the genes and try to find out what is wrong. So we have what we call enzymes, restriction endonucleases, which cuts the DNA at a particular site, restriction endonucleases. So we have what we call restriction sites. So the DNA here are being cut into places at a precise area by restriction endonucleases. Then we also have restriction fragment length polymorphisms. To occur if a mutation renders restriction site unrecognizable to its cognate restriction endonucleases, or alternatively generates a new recognition site. So that is your RFLPs, restriction fragment length polymorphisms. So they are trying to find out on a particular portion of the DNA where you have similar fragments of the nucleotides present. What we have here, this is the polymerase chain reaction. This is very, very common now, no? Your RT-PCR test for COVID. So in order to detect the COVID, they do a reverse from a protein to the RNA. And once they have the RNA, because they are using a reverse transcriptase here, and once they have the RNA, they propagate it through what you call polymerase chain reaction. So you're going to use the polymerase chain reaction test. That's why it takes a long time to do this test for COVID, because your virus is just a protein, and we want to find out what is the RNA of this. So we employ what we call a reverse transcriptase so that we can get the RNA. So what is the sequence of the nucleotides in the RNA? And after that, we propagate it through what we call a polymerase chain reaction. And then we are going to compare it, whether it has the same with the virus. And so we can say that if it is the same, then it is the COVID-19. So you can produce, it can produce thousands of copies of the defined segment of the DNA from a minute quantity of a starting material. So PCR enables medical and biological and forensic scientists to detect the characterized DNA present initially at the levels too low for direct detection. So that is the role of your PCR. Okay. Recombinant DNA provides important tool for studying enzymes. Now we will skip this. We will take this up again when we are into genetics. So you will be able to understand this recombinant DNA. What is meant by that? So these are the recombinant DNA. procedures. Okay. Okay, ribozymes. Ribozymes was first discovered by this person. Sick. It's a very unusual name, no? C-H-E-H, sick. His first name is Thomas. Thomas, where is he? Where is Thomas? Here is Thomas. Thomas sick. So it is just started in 1980, where RNA molecules perform catalytic activity. And so they are enzymes. Before 1980, we always presume that enzymes are protein in nature. But because of this guy, he has now found out that RNA molecules can do catalytic reactions. So we have now two types, protein enzymes and ribozymes. Okay, so we have here, the ribosome is the ultimate ribozyme. The ribosome, which translates the messenger RNA, and the product is a polypeptide. That is your ribosome, an ultimate ribozyme. All the while we have been thinking that there are other enzymes, there are protein enzymes that are involved. But actually now, the ribosome is actually a ribozyme. So for many years, it was assumed that ribosomal RNA, this is the ribosome, played a passive structural role only, or perhaps assisting and recognizing areas of the messenger RNA. But now we have seen that it is actually an enzyme. Why proteins did not take over all catalytic enzymes? Well, as I say, presumably in the case of ribosome, the process was both too complex and too essential to permit much opportunity for possible competitors to gain foothold. In the case of the small self-cleaving, so cutting RNAs, and self-splicing introns. And they may represent one of a few cases in which RNA autocatalysis is more efficient than development of new protein catalyst. As you go deeper into biochemistry, you can see how wonderful is your body being made by your creator. And what we have just accomplished in understanding the functioning of the body we are not yet even 50% of that study. We are too far off. And especially now when you integrate the soul or the spirit, that will become very, very complex. I don't know of your Creator will allow you to understand the workings of the Spirit. Thank you for attending. Do you have questions?